For many microbiological tests, it is important to have a pure culture of that organism. We will choose 2 colonies that were isolated from the river water sample and describe their morphology. See the earlier video on all the different ways to describe colony morphology. These colonies will be restreaked onto selective and differential agar to obtain a pure culture. We will be using McConkey agar and azide blood agar. McConkey agar contains crystal violet to inhibit gram-positive organisms and bile salts to inhibit non-enteric organisms. It also contains lactose and neutral red to differentiate between lactose fermenters and non-fermenters. Fermentation produces acid which turns the indicator yellow. Therefore, McConkey agar is selective for coliforms and differential for lactose fermentation. Azide blood agar contains sodium azide to inhibit gram-negative organisms and defibrinogenated horse blood, which allows different patterns of hemolysis to be differentiated. Alpha hemolysis is the reduction of hemoglobin, which produces a green colour. Beta hemolysis is the complete breakdown of blood, which results in clear zones around the colonies. And gamma hemolysis, or non-hemolysis, does not affect the blood, so the colonies grow on the surface of the blood without affecting the agar colour. First, sterilise an inoculating loop and allow to cool. Next, pick the colony for investigation from the primary isolation plate. Use this to inoculate a small area on a fresh plate. Then re-sterilise the loop. You should first place the loop at the bottom of the flame to render organisms non-viable. Then move the loop up to the hottest part of the flame. This prevents aerosolization of viable organisms onto the workbench. Streak 3 to 5 lines out from the original inoculation site around the edge of the plate. Take care not to touch the edge of the plate, as this may not be sterile. Repeat this procedure, working your way around the plate, making sure to sterilise the loop in between each time. With each set of streaks, the concentration of bacterial cells will decrease, allowing individual colonies to be isolated. This procedure will be performed for both colonies on McConkey, azide blood, and the non-selective yeast extract peptone agar. Proper restreaking will allow individual colonies to be easily picked, as shown here.